this computer. Welcome, everybody. My name is Tommy Soares. I'm the Assistant Director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease. And today I have a pleasure of welcoming Claire Overly from the Nick Noinach Lab in Biological Sciences to our Purdue Lecture Hall series. And we welcome Claire to give us a lecture today and tell us a little bit about herself. I know, Claire, you've been working and Dr. Noinach's lab has been working on some really dangerous gram-negative bacteria, and there's some scary stuff. And I can't wait to hear all about your big microscopes and the microscopic bacteria that you're working on. So welcome to the program, and thank you so much for taking time to talk to us today.
Thank you so much, Dr. Soares. I'm very glad to be here. I'm going to take a minute to share my screen, if that's all right.
For sure. And do I need to allow you? Oh, no, yeah, here you go. You have, great. Nice. So
are slides visible?
Perfect. Excellent.
Great.
I'm going to mute myself and turn myself off. So
okay.
You have the spotlight.
So thank you all for coming and for taking time to be here. I am first going to hide this bar here. And you are welcome, if you feel comfortable, to turn your cameras on if you're interested. I am actually going to ask some attendees some questions, and if you feel comfortable to answer, that would be great. And if not, that is fine too. So as Dr. Soares mentioned, I'm getting my PhD in biology at Purdue. And today I'm going to kind of do a backwards to forwards talk. So first I'm going to tell you what I do, and then I'm going to tell you why I do it. So first of all, I'm kind of interested in hearing what year students are, and if they've taken biology, and if they've ever used a microscope in biology.
So we're here from high school. So they may have just used it in like grade 10 science, and grade 8, we start grade 8 and grade 10.
Great,
no.
So I'm hearing grade 10 science, and they've used some light microscopes. Is that correct?
Yes.
Awesome. So the first thing we're going to do is play a little name that microscope part game. I'm not going to ask you to name all of them. But curious if any of you students know, for instance, what this part of a microscope is. The very top right where you put your eye, usually. We actually have this in our body as well. If you're wearing glasses, you have 2 extra of these. Anybody know what this part of a microscope is called?
Sorry?
I heard someone say something. Do you mind repeating that for me?
A lens.
A lens, that's right, absolutely. The next part is usually how you hold the microscope. You always hold them with 2 hands, as I'm sure you know. We also have these on our bodies, attached to our bodies by shoulders. Anybody?
Arm.
The arm, yes. Okay, something we don't have on our bodies, but usually light microscopes have 2 of them. They help us go from blurry to less blurry to maybe even clear. Anyone know? These are focusing knobs or adjustment knobs. This part highlighted in orange here is where you might put a specimen to look at. This is the theater kids' favorite part of a microscope.
The stage.
The stage, you got it. Okay, the last one I'm going to ask you about usually rotates, has a 3-part of the microscope that rotates and is labeled like 10x or 40x or 100x. It helps magnify. Anyone know? Might have heard it. It's the objective. So there are some more parts to the microscope, and I think you're probably familiar with the concept if you've used one, that there's a light source at the bottom that shines up and illuminates the sample, and then the objectives and the lens kind of work in combination to magnify the image, which you can view at the top by putting your eye into the lens or onto the lens. Okay, so these are light microscopes, but the microscope I'm going to be talking about more today is an electron microscope. And fortunately for us, there are some similarities between these two types of microscopes. So instead of a light source here, we have an electron gun and a high voltage source. And this sends an electron beam all the way through the microscope. You can see it colored here in yellow. There are also lenses on an electron microscope. You can see several sets of them here and the objective lens closer to the bottom. And these are sometimes, rather than glass lenses, they are going to be like magnets. There's not a stage in this microscope, but there is a specimen holder. And rather than viewing the image at the top of the microscope with an eyepiece, there's a screen at the bottom. And so this is, what I'm describing right here is transmission electron microscopy. And so some of the electrons are transmitted or go through the sample, and some of them are deflected by the sample. And all of this information is collected at the very bottom on this screen and is usually sent to a computer that you can view the image on. And there's a few more parts. The aperture means opening. And so in brief, these are some of the parts of an electron microscope. And you might ask yourself, why do we even need this? We have light microscopes. They're easy to come by. They're relatively easy to use. And we can see cells in them without having to do a lot of preparation, which is good because we want to, as biologists, we want to see cells in a native environment or as close to it native as possible at times to understand physiologically relevant ideas about them. So why electrons? And first of all, I want to see, what do you guys know about electrons? Here, I'll show you a picture of electrons. Okay, it's not a picture, it's artwork. We don't have pictures of electrons, but can anyone tell me what they know about an electron? Is it big or small?
Small.
Very, very, very small. Okay, so this is a subatomic particle with a negative charge that orbits the nucleus of an atom. And it has properties of a particle and of a wave. So today we'll take advantage, or we'll talk a little bit about its wave properties. I think it's just very fascinating and able to be used in a lot of ways. I'm not a physicist, but I do think that electrons are cool. So we're using electrons, and these help us out. Why electrons? Because we're looking at very, very, very small things. And so this scale shows us what is available using a light microscope. Can you all see my pointer, my cursor? Let me see if I can get a pointer up. Okay. So if you've used a light microscope, you might have used it, for instance, to look at a plant cell, and you can see the cell walls, and you can sometimes see the nucleus, or you might have looked at like epithelial tissue, skin cells, and again, you can see the nucleus, you can see the boundaries of the cell. Sometimes, if you're looking maybe at pond water, you can see bacteria or small single-celled organisms motoring around in the water, but you really can't get a lot smaller than that. And there's a lot of biology that's smaller than that. In fact, what I'm interested in is something that's about 10 nanometers big. It's a protein that I'll tell you about a little bit later, but I can't look at it with a light microscope. And so that is why it's important to be able to use electron microscopes. And briefly, the reasons we can use electrons to look at small things is because their wavelength is shorter. Higher energy, it allows you to resolve smaller things. So resolution is being able to differentiate between 2 points. And at a lower resolution, those 2 points might look like 1. At a higher resolution, they'll look like 2. And so you can see visible light over here. And then you can see at a certain dosage of electrons, there's a much, much smaller wavelength that allows us to resolve much, much smaller things. So what I use is a type of electron microscopy called cryo-electron microscopy. So you might be thinking that means it's very cold, and you're right, it is very, very cold. And so what we do is we freeze our samples, we store them at really, really cold temperatures, and we collect data on them at really, really cold temperatures. So this is a machine we use to freeze our samples. And I just think this process is really cool. So I'm actually going to show you a video on the next screen showing the process. But the video is kind of zoomed in. And so I want to give you the bigger picture before we watch it. So to start out, what we're going to put our proteins on are these really tiny 3mm grids. And the grids have rows and rows of squares rather, and the squares have rows and rows of very teeny tiny holes. And that is where a protein is going to get stuck when we apply it to the grid. And we're applying microliter amounts. So I'm sure you've used milliliters in lab. We're going to use just fractions of that, microliters of sample, to put on this teeny tiny grid. And then we're going to stick it in liquid ethane. So ethane is usually a gas at room temperature, but this is so, so cold that's a liquid. And we're going to plunge it in so fast that we get a layer of vitreous ice. So you're probably familiar with crystalline ice, like if it's cold outside and you breathe on your scarf or your hair and you get crystals. We don't want crystalline ice, we want vitreous ice, a very, very thin layer. So what we do is we apply the sample to the grid, which is going to be hanging down right about here in this humidified chamber. So we'll put the sample through the side, and then this bowl is going to be filled with liquid nitrogen on the outside, and there's a little teeny tiny metal cup that you'll see that is, it's insulated, so it's not touching the liquid nitrogen. But then we'll put a metal piece that looks like a spider to cool the cup down. So we're indirectly cooling the cup using the liquid nitrogen, which is negative 320 Fahrenheit. I wish I could put that in Celsius as well. I didn't check the Celsius temperature, unfortunately. It's very, very cold.
I think it's minus 196 Celsius.
196. Okay.
I think that's right.
That's helpful. Thank you.
Please correct me if I'm wrong, but I think if my memory serves me well.
Yes, it's very cold, and ethane is actually comparatively warm, but it has slightly different heat properties, which is why we freeze in ethane. Anyway, so I'm so sorry. The lights just went out in my room. But as long as you can still see, we'll continue. So we'll plunge the sample into the ethane, and then we'll move it to liquid nitrogen, which is even colder, and store it in that. Okay, so I am done talking for a second. I'm going to show you a video of this process. So here is the bowl with the insulation. Being filled with liquid nitrogen. And here, I'm going to pause it. So they're actually applying the ethane as a gas, but the little metal cup is so cold that it's going to condense on the spot. this part is the spider that's cooling the cup down indirectly. Now you see liquid ethane, liquid nitrogen, the spider comes out. And this is what we store those tiny 3 millimeter grids in. So that is freezing cold too. So if I pause this, you can't actually see it, but there is one of those grids on the very end of these tweezers. So now the sample is going to come in through this port on the side. And the excess buffer will be blotted off by these little paper circles. And then just like that, the sample is frozen just in that split second. Okay, and so this is the grid going from the liquid ethane into the nitrogen.
Mars Key, please come down to the main office. Mars Key.
And then it is stored in that grid box until the grids are, they're usually prepped for the microscope, and then they go into the electron microscope for data collection. So this is one of the electron microscopes for cryo-EM at Purdue. And it's very, very big. So it has its own room to itself. You can see in the reflection, this is actually the door behind and me trying to take a picture of it. So it's a very large instrument. And if you were to open this door right here to put some sample in, this is the sort of thing you would see. So it really, to me, looks like a time machine. And then there is a set of computers outside that show the images to us. In fact, you can see right here a grid with these rows of squares. In the darker part of the grid is where the ice is thicker, and the part that's a little brighter is where there's thinner ice. And so it's kind of not an exact science, but sort of a game to figure out what exact thickness of ice your sample needs so that you can collect some data. So Purdue has several of these where I am studying, but there are also centers across the country that have lots of electron microscopes, and they allow academic labs to apply for time and collect data there. And sometimes these data collections take overnight or even days. And so several of us from my lab got to go to New York City just a few weeks ago to collect data at one of these centers. This is called National Center for Cryo-EM Access and Training, and CCAT at the New York Structural Biology Center. And so people come from across the country and even internationally to collect. And so we put our pin in Lafayette, Indiana, where we are coming from. And here are some of the microscopes that they have. So this one is very similar to the first one I showed you at Purdue. This one is a slightly different make to get a sense of just how big these things are. And I think I mentioned this, but anytime you transfer the sample, it has to be in liquid nitrogen. So we were here taking samples from those little tiny grid boxes and putting them into a cassette, a little metal rectangle that has slots for grids, which is what the microscope recognizes and what we insert it into the microscope. And then after the microscope recognizes the grids and the lined, these microscopes are a bit high maintenance, but once everything is ready, the microscope takes lots and lots and lots of pictures or micrographs. So down here, you can see the 200 nanometer scale bar. I mentioned a little earlier that my protein is about 10 nanometers. And so you actually can see, if you look really closely, a bunch of 10 millimeter kind of blotches, patches. And this grid is pretty crowded. There's a lot of them. And so lots and lots and lots and lots of data processing later that I'm going to skip. What we see is this gray blob, which doesn't look like a lot, especially in 2D. It's nicer when you have software where you can rotate it in 3D. But what this allows us to do is to fit protein structures in it. Amino acid by amino acid. So if any of you have played with Legos much when you were younger, I like to think of amino acids almost like the Lego blocks that build up each part of the structure of the tower, or the ship, or whatever you're building with Legos. And that's kind of like amino acids and proteins. We need to understand how each part of the protein is built, amino acid by amino acid. So the next thing I'm going to do is talk about the protein, but first I'm going to take a deep breath. You can do that too. You can stretch, smile at the person next to you. And what questions do you have for me?
So Claire, I wonder if you can, just take us, because it's such an incredible thing. If you could just, maybe if you want, I can also help explain in my own Mickey Mouse way why it takes overnight for the microscope to take these thousands and thousands of micrographs. And I want each student who's listening to kind of imagine themselves going with a magnifying glass and walking the size of a football field and taking an image step by step to then recognize which blades of grass are pointing this way versus which blades of grass are pointing that way, and which blades of grass are pointing in all the different orientations. Because what really happens is that when you drop that sample onto the grid and you freeze it, you're hoping that that sample only has that protein that you want to image, because otherwise it's a dirty sample and it has lots of different other proteins that you don't know which is which. So hopefully the set of proteins or the protein that you want to study is the only one there, so it should be fairly pure. But this thing is in 3D space and it rotates, so it's in a very different ways as it froze in many different times. And so this microscope has to go and image and image and image and image until it can take all of these images and say, okay, well, some of them look like they're in this way, some of them look like they're in that way, and some of them look like in that way. And so on average, we really think that if it's in this direction, the protein looks like this. If it's in that direction, the protein looks like that. And so that's why it takes so long for this microscope. And it's like you walking the football field telling us about the direction of the blades of grass. And this is an amazing science that takes a lot of computation, image analysis, as you can imagine. So this field of electron microscopy that Claire is talking about involves a lot of other fields as well, right, Claire? So like what Do you have to know all these other sciences, or are there other people that help you with that?
That's a great question. And I really like the football field blade of grass analogy. Unfortunately, I have a lot of help in understanding what to do with this data. And so I just started learning these microscopes this year. I started in January, actually just with sample prep. And at Purdue, we have a facility. So we have scientists that help us collect the data because the microscopes are so complicated. And then at the risk of sounding like I'm going to say, well, just Google it, there's really nice online tutorials for software. And
Yep.
so what I have done to learn how to process all the data, all the pictures of the blades of grass, is to ask a lot of questions, first of all, be really willing to bug people, and then to watch YouTube videos of people explaining how this data is processed. But when we went to the New York Center for Structural Biology, that was like 3 dedicated days of people teaching us, you press this button next with the microscope and this button next. And so there is a lot of help to be had, whether it's in person or on the internet. And I think one of my favorite parts of science, and I'll mention that in the acknowledgements, is all the people that you come in contact with and the community that science is. So I've made much use of my community in learning these things. And so you made a good point. I'm a biologist. I'm not a physicist, and some of this is complicated physics. And so no, I don't need to understand all of that. And I think, you know, the more you understand, the better. I mean, always never want to stop learning. But it is possible to start these experiments and not know everything and really not know a lot. So that is my plug for electron microscopy. And now I'm going to tell you about my PhD project. And so this protein that I showed you in pink is a protein that's called BAM-A. BAM stands for the beta barrel assembly machinery. And this is a complex. So if you think of machinery, it's a group of proteins working together like a machine that is on the surface of bacteria. And so this is not to scale, but it gives you a little bit of an idea. And it's on the surface of gram-negative bacteria. So can anyone tell me anything about gram-negative bacteria? And before I give you a chance to answer, I don't remember learning lots about bacteria before my college micro class. So I'm curious, have you heard of gram-negatives and gram-positives and gram-staining?
You wouldn't have learned much about it.
Okay, so gram-negative bacteria are, so gram is a stain, and it's basically a way to differentiate between bacteria that can hold the stain. So gram-positive and gram-negatives do not hold the stain. But gram-negatives are characterized by 2 membranes, an inner and an outer membrane. So if any of you in grade school made a cell out of a plastic bag and corn syrup, and I don't remember what, some kind of candy for the nucleus and licorice for the ribosome, this would be like a double bagged cell. It has 2 membranes, and these bugs tend to be really antibiotic resistant. It's just hard to get into them. And so they also have this outer LPS or lipopolysaccharide layer that is also hard to get through. And so the Center for Diseases Control in Atlanta releases antibiotic threat reports from time to time. And in 2019, a number of their urgent threats, or their very top level threats, were gram-negative bacteria. So Acinetobacter and Enterobacteriaceae are bugs that are hospital-acquired infections. This is a huge problem. People go to hospitals for surgery or for some kind of procedure, and then they get an infection at the hospital that does not respond to antibiotics, and they end up sicker than ever. Neisseria also does not have a lot of very good antibiotics against it. Shigella here is not an urgent threat. I think it's a serious threat. So the next level down, but that is also a gram-negative drug-resistant bacteria. And so the reason that my lab is interested in BAM is because it works like a machine on the surface of bacteria, putting other proteins onto the surface. And it's actually essential for survival for these bacteria. So if bacteria don't have BAM, they don't live. And so we are trying to understand how BAM-A is built, sort of piece by piece, so that we or collaborators can understand how to make antibiotics against BAM that can then kill some of these really dangerous bacteria. So my last slide, which is usually my first, is talking about the organism that I work on. So this is an organism called Fusobacterium nucleatum that has BAM-A. And this is an anaerobic bacteria, so its metabolism does not require oxygen. And it lives in the oral biofilm. So one of the really interesting things about our bodies is that any part of our body that is in any sort of contact with the outside has bacteria. So our skin has bacteria on it, our mouths have bacteria, our guts have bacteria. And the mouth has like layers of bacteria. And some of them are healthy, and some of them give you cavities. And Fusobacterium is one of them. It's linked to oral infections, but it's interestingly also linked to early births. That's one of the most commonly isolated organisms from the placenta in women who have given birth preterm. And it's also linked to colorectal cancer, which is kind of unusual for bacteria. So it's an oncobacterium. You think of oncology being the study of cancer. This is a cancer-causing or a bacteria that's linked to cancer. So the reason I study BAM-A with cryo-electron microscopy is to hopefully allow the development of antibiotics against Fusobacterium. So thank you for listening this afternoon. And in my acknowledgement slide, I want to just reiterate the importance of community in science. So this is a picture of my lab. My faculty advisor is here. They drove across the country earlier this year for my wedding. And so they're just really dedicated friends, and I'm thankful for them. I also want to thank the New York Structural Biology Center and NCCAT for teaching me and allowing me to collect data in their facilities. Then my training program that funds my PhD right now is the Drug Discovery and Infectious Disease T32, and thanks to the department and then to the National Institutes of Health for funding. And I am happy to take any more questions you might have.
So Dr. Karish, are there any questions from your classroom that you want us to entertain?
It doesn't look like it. I think they really enjoyed it, but they don't have any questions per se.
Okay, thank you so much. Claire, I do have a few questions. This was awesome, and it's so inspiring to see you do such advanced work in biology, understanding the bacteria, understanding how they're built, and trying to find new ways of combating them and defending, protecting ourselves against them. It looks like a nice family in your lab too, which is really good. Tell us a little bit, if you don't mind, what inspired you to get into this type of work? Why do research in this area?
That's a great question. My high school science teachers are the reason that I majored in science. I was not a kid that was interested in science at all until my 11th grade year. And my parents made me take a physics class, and I didn't want to, but I loved it. It was great. And then I took an advanced biology class my 12th grade year, and signed up as a biochemistry major for college. And I think even in college, I had some doubts, but I was able to do a summer of research at Penn State University. And I think that's when I discovered that research labs are not like teaching labs. So I was really worried about my grades all the time, and labs stressed me out because I felt like if I didn't get the right answer in 2 or 3 hours, I was going to get a C. But research lab is not like that. You are working on a problem, and if you don't figure it out today, then you go home and you come back and you try again tomorrow. And so my junior year of college, it was what solidified the desire to pursue research further. And structural biology is not something I was really familiar with until coming to Purdue. Because I had gone to a smaller teaching school, Purdue really opened my eyes to a lot more possibilities and facilities and resources for science. So I was a first-year PhD student when I started learning structural biology, and kind of falling in love with that.
That's fantastic. And were you scared going from a smaller school to a bigger school? And maybe if okay, just so that the students can hear. And for those of you in Canada, your junior year is like your 3rd year of your undergrad. Your senior year is your 4th year of your undergrad. And when here in America we say I went to college, they're talking about your undergrad years before you get to grad school. So just so that everybody understands a little bit what we're talking about. But if you don't mind taking us back so that everybody can hear, what was it like to decide, first of all, when you went to college, when you started to do your undergrad, how did you decide, oh, geez, I want to study biochemistry? Was it just something that other people were doing and you thought, hey, maybe I'll give that a try? Or was there something that inspired you in reading the catalog for the school you wanted to go to? Like, what was it, Claire? And just so that, because I know the students are thinking, well, I like science, maybe I'll go do something in science, but what? I don't know. And how do you decide? Is it important to decide?
Oof, there's a lot of questions wrapped up in that.
Sorry.
So I don't recommend to others the way I picked my major, but the major was listed in the catalog as not biology, but biochemistry and molecular biology. And it sounded cool. And I thought, I'm going to major in that. That sounds cool. So I really liked the fact that there was some chemistry along with biology, because to me, chemistry makes biology make sense on a molecular level. So there was, I guess, that part of it that was maybe more substantive than, wow, this major sounds cool. But that was part of it, and it does sound cool, and it was really challenging. So I did a lot of studying in college, in university, I should say. And yes, I was terrified to come to Purdue from a small school. I really thought that I was going to get eaten up. And I didn't. So it was a big transition. Some of that is just transition from university to graduate school. It's just a very different feel. Some of it was I had to walk really, really far to get to my classes because it's a big campus. And for me, and this might be different for a lot of other people, but I went to university in the same town where I grew up. So there was I'm in a new place and I don't know anybody part of grad school for me. But yeah, it was very scary, and a lot of personal growth, but I think very, very rewarding, very worth it to come to graduate school, worth figuring out how to overcome the new challenges of research and classes and projects and such.
That's great. And I mean, again, the courage that you must have had to make that decision to come, it shows that at the end, you know, it pays off to have a little bit of that courage and take the leap and just know that, and no matter what you decide, you're going to be okay. There is no wrong decision. There is different paths that you can take, and if that path is not yours, you'll realize that pretty quickly, and you will make the right choice thereafter. But you know, you said something that I think was important also for us to hear. If you don't mind, take us back to that summer where you had that opportunity working in the lab and where you knew, oh, I really like this research stuff. What was it about it? It was in your third year of undergrad. What made you decide, hey, I'm going to go try out to volunteer or get a job at this lab? What was it? And can you take us back a little bit?
Yeah, absolutely. So as I mentioned, I went to a primarily undergraduate institution, and I needed to fulfill a certain number of research credits. And I could do those on campus or off. And so I went ahead and applied to a program that is hosted by some different schools called the Research Experience for Undergraduates. It's funded by the National Science Foundation. And so I applied to them at some different schools, and Penn State is the one that accepted me. And so I moved up there to fulfill research credits. And yeah, it was just a totally different environment. I had never experienced a graduate environment before. Was working with graduate students, was seeing rotation students come through the lab, other undergrads, and talking a lot about research, reading a lot of research for the first time. It really was a brand new environment for me, and a really interesting one, a really fulfilling one.
And did somebody take you under their wing, or was it several people that taught you some techniques, or was it just, you know, a few interactions that you used to kind of get you through that and get you to accelerate in your learning process, I guess?
I guess kind of a combination. So I was assigned to a graduate student, and that is often true as undergraduate or university undergraduate researchers. For instance, now I have an undergrad assigned to me, and I help him through his experiments. So I had that mentor. I also had weekly meetings with our research group, and we would have different professors speak for us. We took a few trips to, at least one trip to a company to see some industry. And I kind of made friends with the rotation students. They were the ones that I had quite a bit of fun with. But there were sort of different levels. I also met with a PI, the principal investigator, the professor of that lab. So there were several avenues through which I received mentorship in that summer.
And I just so everybody knows, these rotation students, these are students that haven't decided yet. They're going into grad school, and they haven't decided yet which lab they would like to go and do their grad program in. So they rotate in maybe, you know, 3 different labs, and they get to decide, but also the principal investigators of those labs get to decide as well, if it's a good fit, if it's not, or if they like it, don't like it, whatever it might be. So that's interesting. So you got closer in with those rotation students, and you said that was at Penn State. So you had to travel away from home and be there the whole summer. Like, were you living in residence and stuff like that?
Yeah,
That's awesome.
I'm from South Carolina, so it was probably about 10-hour drive up to Penn State, and we were living on some on-campus apartments. I think it was the same complex where the football players lived because it was really close to the football building. And yeah, so the group of us, they had a pretty nice cohort there that summer. We lived on campus, we worked on campus, and gave each other rides off campus now and then.
Nice. That's great. And then you said, okay, I'm gonna go to grad school. Did you, in your senior year, do you remember continuing to work in that lab that you volunteered in, or no, that was in Penn State, that was for the program. So did you seek out opportunities in your senior year to work in a lab?
I did do a little bit. There were a few research opportunities available on campus. And so I think for a semester and maybe part of another semester, I worked on a synthesis project through the chemistry department. But it was a few hours a week. It wasn't a lot. I think in hindsight, it's great to do as much as you can do when it comes to research experience. But I did a little research at my home institution, but not a lot.
And how would you say, if somebody is interested in seeking out a research opportunity in their undergrad years, what's the best way that they should go about that?
So probably two steps. One of them is just reading professors' web pages, Googling professors, reading their web pages, seeing what kind of sparks some interest, and then emailing. And sometimes you should email twice or 3 times because people get a lot of emails. And sometimes when they don't email you back, it's not a personal thing. They've just gotten too many that day. And I think sometimes undergrads come to my lab because my advisor was the one that responded. I'm sure that's true in other labs too, where you'll email 5 and 1 will respond. So just kind of seeing what's available and seeing who has capacity to take you at the time and to train you.
Absolutely. And again, I think as you showed, you got to be courageous and go talk to some of these people. And
Yes.
they're humans too. They like people who are interested in the research that they're doing. So you should feel like you can approach them, right? And send them the emails and make sure that they notice you as well. That's great. And Claire, if you were going to do it all over again, if you were going to start from high school and you're preparing to go to college, would there be classes that you would take to help you prepare better? Or would there be classes in your undergrad that you should be thinking about, or that people should be thinking about, that would help them be prepared for a career where they do scientific research?
Yeah. That is a great question, and it's one that can kind of depend on what you are doing. So, for instance, I wish that I'd paid better attention in my university physics class because now I'm doing biophysics. And I will come across terms that I've heard before, and I wish I understood better. So for me, I wish physics, maybe a little bit more calculus. When I was applying to grad school, I actually was interested in applying to chemistry because that is where I had done my research, was Penn State Chemistry Department. But I didn't have, as a biochem and molecular biology major, I didn't have physical chemistry. And that prevented me from applying to a chemistry department. And I think I applied to some biochemistry departments and ended up in a biology department. But there are different course requirements. And at Purdue, they don't do, many departments do not do qualifying exams. And Penn State chemistry, they did do qualifying exams. So I don't have a big sample size for who does qualifying exams and who doesn't. But that can be a factor too, because usually you need, it's advantageous to be passing at least certain ones of those depending on your research. And I wish that I had done a summer of research before my junior year. I think that would have given me a better advantage when it came to applying to graduate school, because that's a big deal for people to look at what you know about being in a lab. Can you pipette? Can you understand experimental processes? Those sorts of things are important for science.
That's wonderful. Claire, thank you so much for taking time to talk to us. I'll open it back up to Dr. Karish and anybody else who might have a question of Claire. And if not, please help me in thanking Claire one more time. Give her a nice round at the classroom. Thank you guys also for joining us. Thank you, Claire. Best wishes for you on your research. It's so cool. It's so advanced. The microscope is awesome. Your work is really inspiring, and I hope that people today also could get a little bit of that. So thank you. Thank you, Claire.
Thank you for having me. Appreciate it.
Absolutely. And thank you all. We'll see you at the next one. I'm going to stop recording now.